Let's see now the active membrane transport. The main thing about active membrane transport that it uses energy. So now we are consuming energy to do the transportation. Why do we need to use energy? Because sometimes we are transporting large particles across the plasma membrane, so we need to spend some energy. And importantly, sometimes we are moving substances or particles against their concentration gradient. Against their concentration gradient. What does it mean? It means you are moving them from areas of flow to high. Remember, in diffusion was from high concentration to low concentration, that is along their concentration gradient. But in active transport, no, it's from area of low concentration to the area of high concentration. And this is against their concentration gradient. For the active transport, we have two examples, either through a pump or the vesicular transport. For the pump, there is several examples. We will see sodium-potassium pump. In order to understand the sodium-potassium pump, very briefly, we need to understand that the cell usually prefers to keep the sodium out and keep the potassium in. This is how the cells would prefer the distribution of sodium and potassium. They prefer most of the sodium to stay outside the cell and most of the potassium to stay inside the cell. Now, this would create a concentration gradient, for example, for the potassium toward outside and a concentration gradient for the sodium toward the inside. And therefore, some of these potassium ions would leak toward outside. The same situation, some of these sodium ions would leak toward inside. So the cell would use a pump. It's called a sodium-potassium pump, as you can see here, to eject these sodiums out and bring the potassium in. So it moved the sodium against its concentration gradient, and at the same time, it moved the potassium against its concentration gradient. It moved it from low to high. And of course, this pump would use ATP. Now, here there is a video that would go over some of the details regarding this pump. So please watch the video. I'm not going to play over through my recording, but please watch it. It's very informative and will help you to understand the sodium-potassium pump. The other two examples of active transport are vesicular transport. These do not involve membrane carrier. The potassium pump, the sodium-potassium pump, if you saw the videos, is basically a facilitated transport that would use ATP. Vesicular transport does not use any of the carriers, no carriers. What happens? Usually it's part of the plasma membrane. Remember, vesicular transport, it's an active transport, it uses ATP. And we have two types, endocytosis and exocytosis. Endocytosis, it means bringing particles in. So what happens that this is the desired particle. Part of the plasma membrane would go around it, will engulf it, and take it inside. Either the membrane would go a little bit outside its border or would form a groove and take it toward inside or through a receptor. It doesn't matter. All have the same concept, that part of the plasma membrane will surround the desired particle and it will pop off and go down. This process would use ATP, and this is endocytosis. It took it inside. Exocytosis is the opposite. So there is a vesicle that contains some of the materials. These could be hormones, could be proteins, could be enzymes, different things. This vesicle would go toward the plasma membrane and it will fuse with the plasma membrane and then it will release these materials outside. It's exactly as you are taking a pill out of its cover or out of its packet. You will pop it off. This is the same concept. This vesicle would pop off or release the contents to the extracellular fluid, again by merging into the plasma membrane. This is exactly the opposite of endocytosis. For endocytosis, this is a process that could be used by some of the white blood cells to deal with bacteria, to take down bacteria. They will engulf them and eat them, or not necessarily bacteria. Sometimes it's bacteria, sometimes it's food, different types of materials, and so on. Another thing that we will talk about and try to understand is the membrane potential. And for the membrane potential, this concept, you will not be able to fully understand it now until we go over it again in the muscle chapters. So that's fine if you don't fully understand it by now. I will tell you what you need to learn from this chapter about the membrane potential now, and leave the rest until we have it in the muscle chapter. For the membrane potential, let's move to one note to do some drawing. So we already talked about that the plasma membrane separates ions. So this is the plasma membrane, right? And we said it separates sodium. It prefers that sodiums would stay outside and potassiums would remain inside. Now, along with the sodium and potassium, of course our body contains other ions, other particles. Some of these particles are positively charged and some of them are negatively charged. Remember, this is outside, this is inside. Now, the net distribution of this charged particle would lead or would give us that outside is more positive comparing to inside, and this is at rest. So if the cell is not doing anything, it means that inside is negative, outside is positive. This is the net charge of all of these charged parts. You can think of this one end that is positive and the other end is negative as a battery, right? The battery has two poles, one positive and one negative. This would lead to polarity, as we saw it in chapter 2, right? So here we have a polarity in the plasma membrane. One is positive and the other is negative. Now, what contributes to have negative inside and positive outside is, at least for us now, what we will focus on is the potassium ions. So for us now, the potassium ions are the one responsible for creating the membrane potential. And to understand this, we will do again a new drawing of the plasma membrane, but this time we will only mention the potassium ions, so no more sodium. We can say that outside is positive and inside is negative, okay, just to keep this in mind. Also, we will draw some of the channels. These are potassium channels. Of course, outside there is maybe one or two potassium ions. We'll put more here, just to indicate that inside there is more potassium ions comparing to outside. Now, if we can think of the potassium ions distribution, we can say that this is an area of high concentration and this is an area of low concentration. And because of this distribution, these potassium ions will try to diffuse through these channels toward the area of low concentration. So they will come and gather around the gate, trying to push it, to open it. Think of this as like the Black Friday night when people are trying to get into the stores. They will push against the gate. Sometimes the gate will open and a few people would get inside, right? In this case, the potassium ions would push against the gate and some of the potassium ions would be able to leak outside. We understand and we talked that the cell would prefer these potassium ions to be inside. So what will happen is that we can think of the sodium-potassium bump that would bring these potassium ions back using energy. So at one side, there is a movement of potassium ions toward outside, and at the other side, there is a movement of potassium ions toward inside. Both movements will happen at the same time. Once we have equal rate of potassium leaving and entering the cell establishes its membrane potential. So once the rate of potassium ions or the number of potassium ions leaving equal number of potassium ions coming back, then we will have the membrane potential. Now, this does not mean that the number of potassium ions across the plasma membrane will be equal. So we will not have like here 10 and here is 10. No, this will not happen. We're talking about the rate of diffusion out and transporting in. Once this rate is equal, then we will have the membrane potential. You can think of this as if you have a water tank. There is a water tap in this tank causing water to come out, and there is also a water tap or a water source here. So if we have the same rate of water coming in and water coming out, then we will have a stable level of water within this tank. So this is exactly the same. If we have same amount of potassium ions leaving and same amount of potassium ions coming out. So we will have a stable potassium ion concentration that would keep inside negative and outside positive. That is when the cell would establish its membrane potential. So what you need to understand that the cell has a membrane potential. This membrane potential means that outside is positive and inside is negative, and it is established when the rate of potassium ions diffusing outside equal the rate of potassium ions transported toward inside. And lastly, that there is, in this process to establish the membrane potential, we have diffusion and we have active transport. Diffusion was here and active transport was here. Now, in the slides, it will tell you, it says that the potassium coming back because there is more negative charge here. If you want to learn it in that way, I'm fine with this. If you want to learn it in the way I explain it here, I'm fine with this. However, sodium-potassium bump is more accurate than the contribution of the negative charge on the inner side of the plasma membrane. So I will stop here, take a break, write down your questions, try to recap the membrane potential. As I said earlier, you will not be able to fully understand it until we see its applications in chapter 9 and chapter 11.